Hello, today I'm going to talk about the localization of the OZ proteins. But first I'd like to give a brief introduction about RNA editing, because one of the OZ proteins is involved in the RNA editing process. RNA editing occurs in flowering plants within the organelles. Excuse me, I'm sorry. C is converted into U within the transcript of the RNA. In Arabidopsis, there's 43 edited Cs, whereas in the mitochondria, there's 619 edited Cs. The correct C target is identified by a cis element. For different C targets, there's different cis elements. The RNA binding protein pentatricopeptide repeat, or PPR protein, recognizes the different cis elements. There's also different PPR proteins to complement the cis elements. Further research done in the Hansen lab identified additional proteins present in the RNA editosome, including ORM, OZ, and RIP. Each type of protein present in the RNA editosome is a member of the protein family. In Arabidopsis, the PPR protein consists of more than 400 members, whereas the RIP protein family has 10 members, ORM protein family has 30, and OZ protein family has 4. It's important to understand that when C is converted into U in the editosome, there's a variation between the proteins present. So in other words, depending on what C target there is, the proteins in the editosome can differ. OZ1 was found to associate with previously found ORM1 protein, as I mentioned. Knockout experimentation demonstrated that the effectivity of RNA editing various C targets was affected. The localization was found to be in the chloroplast for the OZ1 protein. These are the chloroplasts. Normally they fluoresce red due to the autofluorescence of chlorophyll B, but for this presentation I decided to show them as blue because I thought that it would ease the viewing process. This is a YFP tagged OZ1 construct. You can see a little bit of punctated interaction. This is where the protein's interacting with the nucleoid. And then here we have an overlap of the two images. Excuse me. So why do the OZ proteins, what are the OZ proteins, I should say? There are two domains that are shared by the members of the OZ family. One consists of multiple highly conserved regions at the N-terminus, represented by the red, blue, and purple boxes. The other is a RAN-BP2 type zinc finger. At the C-terminus, there's more variability. There's also variability in the number of zinc finger motifs present. OZ1 and 2 each have 2 zinc finger motifs, whereas OZ3 has 3, and OZ4 has 4. So RAN-BP2 zinc finger proteins bind RNA. Plant organelles use RNA binding proteins to control various processes, including RNA stability, RNA cleavage, translation, transcription, RNA splicing, and RNA editing. So the question is, which of these processes do the OZ family proteins affect? And to better understand this question, we need to understand which organelles the OZ family proteins are located in. We don't know a lot about the OZ family, but the member of the family that we know the most about is OZ1. The OZ1 mutant exhibits impaired editing at 30 chloroplast C targets. And as you can see, there's impaired phenotype. For OZ2, the homozygous mutant is embryo-lethal, so we have not been able to study this protein in detail. However, for OZ3 and 4, there's no variation between the mutant phenotype and the wild-type phenotype. So this raises the question of what are these proteins doing that's causing such a dramatic variance in phenotype of proteins within the same family? And increases the question of what's the function of these proteins? But first, to understand these fundamental questions, we need to understand what the subcellular localization of the proteins are. So as a start, I used a bioinformatics program, Target P, that predicts the localization of proteins. It does this by analyzing the N-terminal region of the protein, which contains the transit peptide, which is responsible for targeting the correct organelle that the protein will be located in. So the program produces a prediction score which is shown in the graph. For OZ1, 2, and 4, it predicted the localization to be within the chloroplast, whereas for OZ3, it predicted the localization to be in the mitochondria. So I used Arabidopsis cDNA and then amplified the protein sequence of interest using PCR. I then did a TA cloning reaction with the linearized vector PCR8, then did an LR gateway cloning reaction with the destination vector PEX-SG-YFP. Then using the plasmids, I transformed Agrobacterium and then did agroinfiltration on the leaves of Nicotiana benthamiana. I also transfected the protoplast of benthamiana and the protoplast of Arabidopsis. All three experiments were observed using fluorescent microscopy. These are my fluorescent controls for the protoplast. Here you can see there's not much expression, there's no expression on the YFP channel or no signal. I also included a control without a stained mitochondria and a mitochondria that I stained using MitoTracker Orange. Once again, these are chloroplasts, normally red, but I'm showing them in blue to ease how we view them. And then the last image is an overlap of the previous images. I'll be using a format similar to this for the remainder of my presentation. I'm actually going to dim the lights a little bit more so we can see. So these are protoplasts from Arabidopsis. Here we can see the protoplast is expressing the OZ2 construct that I tagged with YFP, shown in green. Then we can see the stained mitochondria, and then when we combine these images, we get an overlap of the two, where we can see a trend of co-localization between the mitochondria and the OZ2 protein. Additionally, I have data from the agroinfiltration. So this is an image of a leaf fluorescing my protein in green, and we're viewing the leaf from the bottom. Then we combine the chloroplast and the protein to get this image, and we can see a similar trend of there's not co-localization between the protein and the chloroplast. To make this easier to visualize, I rotated the image so that we're viewing the leaf from the side. Here we can see there's no trend here of co-localization between the chloroplast and the OZ2 protein. We can also see that here. In addition, I have protoplasts from Nicotiana benthamiana. Here we can see the mitochondria, and this channel is only expressing the chloroplast and mitochondria, whereas this is also expressing the protein. And we can see a strong trend in dual localization between the mitochondria and the protein, and from this we can conclude that the OZ2 protein is localized within the mitochondria. Next, I'm going to talk about OZ3. So OZ3, we have a similar setup, expressing the protein in green and stained mitochondria in red. When we overlap the pictures, we can see a trend of dual localization between the mitochondria and the protein. Looking at the agroinfiltration data, we can see, once again, a trend showing that there's not really a relationship between dual localization of the chloroplast and the YFP tagged OZ3. Rotating the image and looking at the bottom image, the leaf from the side, we can see much clearer that the protein is actually fluorescing in what seems to be the shape of the mitochondria. Looking at the protoplast from benthamiana, we have the red mitochondria, and then here the protein is being shown to fluoresce in this orange-yellow color. This is because it's overlapping with the red and the green. And from here, we can also conclude localization within the mitochondria for OZ3. This next protein is the hardest one for me to determine the localization, so I included an extra image of only the YFP tagged OZ2 channel. Here, you can see there's spots of localization where the chloroplast is not present. And when we compare this to the chloroplast and mitochondria overlap, we can see similarities of where the spots are located. I've also included an overlap of all three images where we can see the orange-yellow fluorescence in the mitochondria, showing that the protein is indeed being expressed within the mitochondria. Additionally, we have fluorescence coming from the chloroplast, and they're actually occurring in this punctated fashion. So in review, I found the localization for OZ2 to be in the mitochondria, which is inconsistent with the bioinformatics program target P. I found the localization for OZ3 to be in the mitochondria, which is consistent with the target P prediction program. And for OZ4, I found dual localization with a favor and distribution for the chloroplast, which is consistent with the prediction program. So how does the localization of the OZ proteins inform us about their function? It specifies the extent at which the OZ family is distributed between the two organelles, and identification of the OZ protein localization will focus RNA processing analysis on proper organelle OZ mutants. In summary, I would like to thank everybody that's made this experiment possible, including NSF, BTI, and Cornell University. Cornell University Biotechnology Resource Center, specifically the Imaging Center for letting me use their microscopes. I'd like to thank everybody in the Hansen Lab for making the summer enjoyable and helping me and supporting me along the way. But I'd like to give a special thanks to Andrew, my mentor, Maureen, and Stefan. They've been so much help over the summer, and it's been so good. If there's any questions, I'd be happy to answer them. Thank you. I actually did a co-infiltration with another construct, P19, and this helped to prolong the expression of my constructs. So what I did is I made an infiltration medium at which then I would scratch the bottom of the Nicotiana leaves, and then without a needle inject the bacteria-containing solution into the leaves.
So do you have a hypothesis for why the target P predictions were wrong?
I do not have a hypothesis for that. I don't understand the way that the program comes up with the predictions well enough to be able to conclude a hypothesis.
Do you have any plans to make double mutants to see if there are 3 or 4 double mutants?
As far as I know, I'm not sure. I know that there's been a little bit of talk about maybe there's a little bit of redundancy between the proteins, and that might be why the phenotype isn't being affected as much in OZ4, because maybe OZ1 maybe OZ1 is taking up the slack that OZ3 or 4 isn't really working type of thing.
So maybe one more quick question back there.
Do your experimental results clarify the ambiguity prior in the literature, or does it help guide some future questions?
I'm not completely sure what you're asking, but I would say I have a lot more data than what's shown here. I actually did the experiment 5 times, and I'm getting concise results that are consistent within each other. So I would say that yes, it does clarify the ambiguity.
But does that localization help contribute to your knowledge of what the functions of those are?
Oh, yes, definitely. By determining the localization of these proteins, we can better target where we're going to focus our analysis of RNA processes, because then we can either cancel out one organelle that we're not going to look at. So for, say, OZ2 and OZ3, we don't need to look within the chloroplast for RNA analysis, which, if you remember at the beginning slide, the mitochondria has over 619 editing sites. So if we can count those out, then we only have to worry about the 30 or so that's within the chloroplast.
Okay, so I think we've got to move on.
Thank you.